Well, my name is Jeremy Poole. I made a mistake already. There we go. Now this title has quite a lot of explaining to do before we start, so I think I'll spend about a minute describing what these words mean. And I'm sorry I'm speaking in English, but I'm sure your English is a lot better than my Czech. I come from the UK. So we agreed on this title, The Art of SEM. SEM stands for Scanning Electron Microscope. And this is the only time I will use the word scanning electron microscope because it's much too long. I just say SEM or SEM. Now, the microscope obviously makes pictures. What I say the microscope actually makes is images. And then it is up to the artist or however he describes himself to change these images into pictures. So I like to make scientific images, but I like to make them look a bit prettier. And I'll explain how I do that. And this is from fantasy to reality in half a century. Now, I wanted an electron microscope when I was aged 19, which was a long time ago. And I shall explain how I changed from someone at the age of 19 who wanted an electron microscope to being the proud owner of a test scan microscope since 2020, I think. Now, I should say my name's Jeremy Poole. I'm actually not a biologist, although all the pictures will be of biological subjects. I'm an engineer, but I've always had a love of the natural world, and it seemed natural to me that I should make pictures of insects and things like that, and there's a special reason I like insects, which I'll come to. I'm also a great fan of spiders. I know a lot of people don't like spiders, but I love spiders. Sorry if you get uncomfortable with my pictures of spiders. So what I'm going to talk about is 6 milestones, which is paths in my life that were major paths towards becoming an electron microscopist. So milestone number 1, I already mentioned a little bit. I first saw a scanning electron microscope, and gosh, I really wanted one. This is what it looked like. This is a Cambridge stereo scan Mark I. It was invented or manufactured in 1965. I saw this in 1968. It was producing a picture which looked something like this. It was green, but it was green because that was the color of the screen on which it was displayed. And it was a small screen here. So we were looking at this little screen and saw in this slightly out of focus green object, which was the eye of an insect. And I just thought this was absolutely amazing. So I said to the professor who was demonstrating it, "How much does one of those cost?" And he said, "That will be £100,000. That's in Czech currency is about 2.75 million. That was an awful lot of money. 3 years later, when I left university, my first salary was less than £1,000. So I thought, well, that's a fantasy which I shall never get to realize. Moving on a bit to 1998, I bought my first stereo microscope. That's an ordinary light microscope used for looking at things like insects, whole insects, at relatively low magnification. And I got to look at spiders, and I like spiders. So let's look at pictures of spiders. So these are what we call photomicrographs. They're photographs taken through a microscope. They are a light microscope. And what these are this one over here on your left is the head of the big house spider, the one that crawls around your houses. And you can see it's got, well, you probably can't see all 8 eyes, but it has 8 eyes. And on your right, that is actually the female sexual organ of the spider. It may look like a male sexual organ, but it's actually female. And I'm going to have to talk a lot about sexual organs of spiders because they are very important, not only for reproduction, but also for identification of the spiders and of species. Right, so that's 2 milestones. Third milestone, I read a review of a book of electron micrographs. So I thought, I'll buy that book. Then I happened to meet the author. He gave a lecture. He was a member of our microscope club I was in, and he gave a lecture on little creepy crawlies that he'd made images of with his microscope. His electron microscope. The electron microscope that he had in a shed in his garden. And I said, how much does an electron microscope cost? And he said, oh, about £50,000 to £100,000. And this, of course, was in 2013. So from 1968 to 2013, the price has not increased at all. This is the book that I read the review of, and that's the title page. And over here is the head of a crane fly. Now, I need to say very clearly that the electron microscope does not give coloured images. These images have been coloured by hand in a program such as Photoshop. And in fact, my friend, who he became a friend, he had a friend of his who made the colouring, so he did not colour them. And I don't colour my photographs, my electron micrographs, partly because I prefer to make electron micrographs rather than spend all my time using Photoshop, and partly because I think they actually look pretty good in monochrome, in black and white. Now, one thing you'll notice about this is it appears to be all in focus from the front to the back. And anyone who has tried to take pictures of insects with a close-up lens will know how difficult it is to get the whole picture in focus. But the scanning electron microscope has an incredible depth of field, as we say, so you can focus over quite a large relative area before it goes out of focus. And I just want to illustrate that with this picture. Here we have a very small shell. It's about 1 millimeter long, and these pictures were taken with a light microscope. And you can see on the left-hand side there's a little bit here that's in focus, and there's a tiny bit here that's in focus. And then on this one I changed the focus, and it's a little bit in focus here, but everywhere else is out of focus. Now if you put it in an electron microscope, you get that. And there's several things you notice about that. The first is it's all in focus. The second thing is it's in black and white. And the third thing is, and I'll come to this later, is that it appears to be illuminated from the top. So at the top it's brightness, and towards the bottom it's in shadow. And that gives a nice pictorial effect, a sort of modelling effect. In fact, some people would say it's a bit like 3D. And there's one reason that the first electron microscope was called the stereo scan, even though it's not at all stereoscopic. It appears to be stereoscopic. So that's one of the big advantages of a scanning electron microscope, is the depth of field. Right, so I now knew I wanted an electron microscope, and I knew they didn't cost, well, they cost a lot of money, but they didn't cost an awful lot of money. And I went to this conference. Every 2 years in Manchester they have an enormous conference and exhibition, and there's a big exhibition hall where people like Tescan and Thermo Fisher and Zeiss and all the other manufacturers bring their electron microscopes, and the other light microscope manufacturers come too. And there I had a demonstration of something called the Zeiss Sigma, which is a very expensive one. I could never have afforded that one. And the Phenom Pure, which is a little one that looks like a coffee machine. It's about this big. And that was nearer my price range, so I thought about possibly buying that. And the other thing is I met I met some people who sell specimen preparation equipment. Now, it's one thing to have an electron microscope, but it's no use at all if you can't prepare your insects to put into it. And preparation, that's a subject in itself, but you have to dry the thing and you have to coat it with gold and various things like that. So it's not straightforward. Now, I said you have to coat it with gold, but I was talking to the Zeiss demonstrator, and we'd established that I could never afford his microscope. He said, "What's your interest?" I said, "I like insects." So he said, "OK, go into Manchester in the lunch break, come back with an insect, and we'll put it in the microscope, and we'll see what we can do." And this is the image he came up with. So with absolutely no preparation, he managed to produce an image of an insect eye. I love insect eyes. And that's what it was. And I thought, great, I don't even need to buy the preparation equipment. But I did in the end. So I was really enthused, and I was determined to buy one. And so I did. The trouble with an SEM is it's too big to fit in the house. So even if I had room in the house, it wouldn't go through the front door. So I had to have a laboratory built in the garden. So over the months it grew, and then it became a laboratory. And there, it's not a test scan instrument. I am on my second SEM now. This is my first SEM, and this one is made by Thermo Fisher. And you know about Thermo Fisher, they also work in test scan in Brno. And it was a very good microscope of its type. This was a second-hand one, that is pre-owned, used and abused by the people who used it before. And of course it's FEI then, but it was now of course Thermo Fisher. So this was January 2016 that I had my first electron microscope. And I'm very ashamed, but I thought I'd have to show you my first image. And yes, it is awful. Not only is it out of focus and the contrast is very bad, but there's dirt everywhere. Okay, it's a soil mite, so it's got soil on it. But I was very depressed when I saw that. But a couple of weeks later, literally 12 days later, I produced something like that. That is the rear end of a spider. It is where the silk is spun from. So there's 1, 2, 3, 4, 5, 6 spinnerets, as they're called. And that's the anus. And I was quite pleased with that picture, really. And once I'd made the image, I wanted to make it better. So that is actually, I post-process it a little in Photoshop. But just as I was learning how to use my electron microscope, I was also learning how to use Photoshop. So it's an early example. 19 months later, and I'm still with the Thermo Fisher microscope, I managed to get one of my images in the calendar of the Royal Microscopical Society. So I was very pleased with that. That's the image. It's a spider head. And this one's got 8 eyes. There's a little one just there. 3, 4, 5, 6, 7, 8. And of course, there's the page from the calendar just to prove it did go in the calendar. So needless to say, I was very pleased with that. Right, so the last milestone in this journey, the last major milestone, I think this day today is my 7th milestone. But the 6th milestone was to replace my Thermo Fisher with a new one. So I went along to Zeiss in Cambridge because I knew Zeiss, I knew the demonstrators, very good company and very good SEMs. And I went to discuss the Zeiss Evo. The Zeiss Evo is their entry level, as we would call it, SEM. And I had spoken to Tescan about their entry level SEM, which was the Vega. But they said, oh, we can do better than that. So they said, how about Tescan Mara? And that is basically one step higher than the Zeiss Evo, or indeed the Thermo Fisher Prisma. So I said, well, why not, if I can afford it? Well, 2 days later, I placed an order because, yes, definitely, I wanted one. Then on the 26th of March, coronavirus hit. Well, I think Tescan knew that coronavirus was coming along, and they needed to give me a really good price because there weren't going to be any other customers for a year or two because all the universities were shutting down and so on. So I blame my good fortune of having a Mara partly on the coronavirus, which is about the only good thing I could say about the coronavirus. And so I placed the order in March, and it was delivered in July, and 2 members of the TestScan UK team came along and installed it and gave me some initial training and left me to get on with it. And this is it. This is my baby. And I'm still very pleased with it. I'm loving using it. And I've made thousands of images since then, of which you will see just a small portion. And just to remind you, whoops, that's what the old one looked like. And you can see there's very little comparison. I mean, this is even artistic. I mean, that is not artistic unless you're into retro engineering or something. But this definitely is artistic. This is a picture of Rembrandt van Rijn, the great Dutch portraitist. And in fact, it's a picture of him by him. It is a self-portrait. Now, I put this picture in because it illustrates what I mentioned when I showed you that picture of the shell, how he lights his pictures. So he's got light that's coming in this way, illuminates this side of his face, and the other side of his nose in shadow, and down here you have shadow. It's basically, in photography, it's called Rembrandt lighting to this day. So it's basically illuminating your subject from above and to one side. And why do we do that? Well, the sun is above us, and it's often at one side coming in through a window. So it's basically how you would naturally see a person. I mean, imagine what it'd be like if the sun was coming in that way. You'd look like this. And frankly, that's not very artistic. So that leads me to the only technical bit that I'm going to talk about, and I should try and keep it very simple, because basically there is a detector on any scanning electron microscope called the secondary electron detector. And this is what it looks like on my Mara. And there is well, it's not a theory, it's a fact, that if you take an image with this detector, it looks as though, basically, if the observer was in the direction of the electron beam, which is basically coming down from the top, the position of the illumination of the picture appears to be where the subject is in relation to this detector. So on many SEMs, the detector is at the top of the image, and that's where it stays. And people will always leave it at the top of the image, and you will always get illumination from the top. Now, you can't actually move the detector. It's firmly bolted to the outside of the microscope. But you can move the specimen inside the microscope. And you can arrange for the top of the specimen to be where you want. So what I'm going to show you next is 2 pictures of the same foram, which is a tiny little shell. One is illuminated from the left. Illuminated is the wrong word. That is where the secondary electron detector is. And this one has the secondary detector on the right. There's not a great deal of difference, but it's just, I just much prefer that one. I don't know about you, but I just prefer that. But most of the time, and I did this for years myself, you just leave the detector up there and never think about anything else. But I only made this discovery about a year ago, that by setting everything up so that the detector is apparently over to the left like that, you just get a bit more artistic. It's not changing anything. It's not making the image any less real or any less scientific. It's just making it look prettier. Now, talking about making images look prettier, when they come out of the electron microscope, they do leave something to be desired, and that even includes with the Mara. Sorry. But we can do things about it. So this is another of these little shells straight out of the Mara. And you can see there's a number of things that perhaps would make you think twice before you wanted to hang it on your wall as a picture. It has these little cracks. These are on the way you mount them, and these cracks sometimes appear, and they're very off-putting. The contrast level is a bit low. It has to be for various reasons, so I won't go into. It's got all this rubbish at the bottom. Well, it's not rubbish to an electron microscopist, but it probably doesn't mean a lot to many of you. The one thing at the bottom you may think is useful is an indication of the overall size. So, how do you transform that into a picture? Remember, I said you start with an image and go to a picture. So, in 2 minutes, you can end up with a picture like that. I call it a picture because it's a picture. Basically, what I'm doing it electronically, but basically I'm just cutting around the outline of this. I'm sticking it on a piece of black cardboard, and somehow or other I'm making it more contrasty, which you can do in Photoshop quite easily. And the other thing I've done is moved this scale bar and put it up there out the way. Now some people say, well, if you're going to hang it on the wall as a picture, why don't you put a scale bar? And well, I have long discussions about this. In fact, I had discussions between the two organisers of this very meeting as to whether you should or should not have a scale bar. So what does a scale bar tell us? It provides an indication of scale. And if you're going to enter your images or pictures into scientific publications or competitions that are connected with science, the organisers, the judges will want to know how big the object they're looking at is. And basically, one way of doing it is to include a scale bar. And a good reason for that is if you then enlarge it and print it big, the scale bar grows at the same time, so you always have the right indication. You don't use the magnification setting on the microscope because every time you change the size, you actually have a different magnification. But as I said, some people might say that as a picture, it's not very good because you've got this technical detail in it. There are other ways of dealing with it. You can just say how wide the whole picture is, and then people can make their own discussion, their own deduction. Right, I've just got a couple more examples of processing that I've done. This is a wasp head, and the processing with this one took me 5 hours. So 2 minutes for the first one, 5 hours for the second one. If you're imagining cutting out the outline of this wasp, you think of all cutting out around all those hairs. So literally I had to do what's called a selection in Photoshop. For each of these hairs I had to separate it from the background. And there's this big blob of glue there. So it took a lot of work, but I was glad I did it because I thought it looks quite good when it's done. That scale bar does get a bit in the way because it's right up against the image, and there wasn't anywhere else to put it. So basically it is quite easy to take it out. And there is the picture. This is the head of an ant, as I said, right? No, this is the head of a wasp, I beg your pardon. We now come to the head of an ant. Again, that's how it came out of the microscope. And that's how I initially processed it. But I thought, yeah, but it's all sort of collapsed around here. It's very easy for the bodies of insects to shrink when you process them. So I thought, what can I do to rescue this? And basically, I did that. So I turned the head around a little bit. I cut off the body altogether. And as you can see, I've taken out the scale bar. And that is one of my favorite pictures. So this is me working on my own at home. But sometimes I get asked to make electron micrographs for other people, because frankly, anyone who knows I've got an electron microscope really wants to have me do things for them. So another outcome of COVID was we had a little project between two of our microscope clubs. And we said, we want to take as many images of this insect, the green shield bug, or it's called Palomina paracina in Latin. It's also called a stink bug for reasons that will come clear. So they said, right, let's look at this from beginning to end of the life cycle, from the eggs to the adult, and see what it looks like. So we start with the eggs, and there's two things there to see. One is they have these little triangular egg bursters. So the nymphs inside, the hatching bugs, will use these egg bursters to escape from. They'll make this hole basically escape from the egg. The other thing you'll see is these aeromicropilar processes. What they are is there's 30 of them around the outside of the aperture, and they have a particular function. In fact, they have two functions. One is they act as aerators or air conditioners for the interior of the egg. And second is they act as a conduit for the sperm of the adult male. So the male will fertilize the egg by placing its sperm on the outside of the egg, and the sperm will migrate through these processes. Another thing to notice is these processes here, these are all designed for preventing insects, or, well, not insects, birds particularly, from eating the eggs. They're just meant to put them off. Right. So the thing about the life cycle of the shield bug is it starts as an egg, as insects always do. Then it goes through 5 stages of growing, and each stage is called an instar or a nymph. So they're not adults, they are babies, and they molt. They shed their skin and grow 5 times before becoming an adult. These are actually pictures of the last instar, and that is the dorsal view or the back view, and that is the front view. I've cut the legs off, otherwise they get in the way. So you can just cut them off, they're dead after all. And the reason I put that picture up is it's the only picture I have of the whole bug. But I've got something called O1, O2, and O3, and L. There are 4 L's and 3 O's, and I'll deal with each of those in turn. L stands for lanceolate. Lanceolate is just basically the shape of a spearhead, so it's just a description of the shape. And this is what it looks like. Now, when we first saw this, we thought, is it a bit of damage? Something happened when it molted. But then we thought, on every nymph we looked at, there were 4, and they were in the same place, so it can't be damage, even though that looks a bit jagged. When we looked inside, we saw this. And that is definitely got a function. It looks like, well, it could be a bit of lung, or it could be a bit of an intestine. We just hadn't a clue. So I said, I know someone at the Natural History Museum in South Kensington, London. She's a spider expert, but she can put me in touch with a bug expert. So I got in touch with the bug expert, and he said, I don't know, I'll consult my friend in Prague. So he's put me in touch with his friend in Prague. And basically, he didn't know, and he's never come back. I think he's probably doing some research in his own now, so he can take the credit. No, that's not fair. But anyhow, so the purpose of this is still a mystery. When these turn into adults, this opening disappears. But for all one of the 5 instars, the 5 stages between egg and adult, they're there. And they're also there on other species of the same general family. These are osteoles. Now, osteole, strictly, is a tube that takes scent from a scent gland inside the insect or its instar and takes it to the outside world. We've used the term a bit more loosely to include the area around the outside. So this is one of those three areas on the back of the bug, as I showed you earlier. And what happens here is, if the bug is disturbed, it will send scent. Well, I say scent, it's not the sort of scent you'd wear, it's the sort of scent you'd run away from. A stink onto the outside and hopefully deter the bird or whatever is trying to eat it from attacking it. And around here you can see there's a whole area which I've magnified here that basically has these little sort of receptacles and lots of grooves and things, and the purpose of that is to make the scent stay in the places. It sticks there with surface tension, and so it doesn't get rubbed off. And when it becomes an adult, this is the amazing thing, that between the fifth instar and the adult, a whole load of things change. And one of the reasons they change is because it's that stage that the adult when it becomes an adult, the adult grows wings, and it needs all that area on the back to house its wings. So the osteoles, or the scent glands, scent tubes, migrate to the underside of the adult, the ventral side, as we say in scientific terms. And on the underside you have a tube that comes out, which is where the scent's sprayed from. And then you have all this area around it. And here is the area in higher magnification. And you'll note I've still got a scale bar there. But I love this picture. It's one of my favorite images because basically it's not obvious what it is. It's got lovely patterning. It's even got something that looks like a flower. Another one over there, another one there. So it's a mystery. But the other things I like about it is it shows light and dark, shows this modeling effect that I was talking about earlier. And I just love it. So I just thought I'd show you a bigger version of it. So when the scent is basically sprayed out of the tube you saw in the earlier picture, it lands on this area. And my theory is that these individual things, they're like mushrooms upside down. So they will retain the scent and stop it being rubbed off when it walks through leaves and things. That's my theory, but I'm no biologist. Another thing that happens when the fifth instar becomes an adult is that there's subtle changes to the head, or more particularly the eyes. So this is the last instar, or nymph, and it's got two eyes. It's got one there and another there, and you see it's got the same compound eye construction that I showed you in that very first image on the Cambridge electron microscope. Here the eyes are a bit better developed, but also it has grown two more eyes, one there and one there. Those eyes are called ocelli. They're simple eyes. Now a compound eye has all these facets, and each facet is a separate eye. These are much simpler, but the theory is that these are used in flight for navigation. And of course, since the instars don't fly, they don't really need ocelli, but the adult does fly, and it grows those. And the whole picture of the adult looks more mature, better shaped, less sort of floppy, in my view. Now, I've mentioned these bugs molting, and molting means they shed their skin, and miraculously what is under the skin then swells up and becomes bigger than the previous instar in this case was before. And this is actually a shed skin. Now the skin is very, very thin when it's shed, but it's interesting to note just how much detail there is in the skin. So you can see the facets of the compound eye. You can see all these tiny hairs everywhere, and you can see the antennae and this thing whose name I've temporarily forgotten. It's just amazing the detail you see on this bit of skin. Anyhow, so that was a joint project I did with 4 or 5 friends, and I really love working in collaboration with other people. I'm fine working on my own. Almost all my spider work I do on my own, but it's always good to have another outlook on an idea. It's also nice to have someone to provide all the specimens in this case, because in order to follow the instars through every stage and know that we have the first, the second, third, fourth, he bred them all from eggs and followed the life cycle through, so we knew exactly which stage of the life cycle we were at. But now, spiders, my favorite subject. I'm sure most of you know this, but spiders are not insects. They're called arachnids. They have 8 legs, whereas insects have 6, and they usually have 8 simple eyes. So simple, that means they're not like insects where you have all these little facets. It means they just have one facet. Like us humans have simple eyes. They're very complicated, but they're called simple because they don't have facets. They're often hairy, and that is one thing that really puts a lot of people off. And many people really do hate spiders, which really makes me angry because they're lovely. As I say, I love them. And frankly, one of the reasons I bought my SEM in the first place was I was getting into spiders. I was looking at them through my ordinary light microscope, and I just couldn't get the detail I wanted, especially in the sexual organs. And I keep going back to sexual organs, but that is the way one differentiates between species of spider. And from my point of view, they are really beautiful, and I don't know whether at the end of this lecture you will agree with me, but I'll try and convince you. But we'll start with the head. I said spiders usually have 8 eyes. Most of the British spiders do, but there are a few who only have 6. And here's an example of a spider with 6 eyes. It's quite a small spider. And all spiders also have what's called chelicerae, which are these, they're basically like jaws. And at the base of the chelicerae they have fangs. This spider has 8 eyes. They've shrunk a bit in preparation, I'm afraid. So 1, 2, 3, 4, 5, 6, 7, 8. This particular family of spiders has a large area between the bottom of the spines and the end of the head there, which helps in identification. The other thing to notice about this one is it's clear that it's got these enormous fangs, and it also has teeth. So what happens when a spider catches its prey, which is almost always insectivorous, it can be things like woodlice and so on, is it basically penetrates the insect with these jaws and pumps venom in, and then it pumps in enzymes and things to digest the contents of the insect inside the insect, and then it sucks the liquid that it's injected, plus of course the insides of the insect through a mouth which you can't really see. It's sort of lurking up there. It doesn't masticate, i.e. it doesn't bite or chew, it just sucks liquid food. So those are pretty typical spider heads. Here are some that are far from typical. I love these just because they are so ridiculous, really. They're both of the family called Linyphidae, which are the tiny little spiders that sometimes land on you when you're not expecting it and sort of dangle on a silk thread from your hat or from your hair. So these are about 2 millimeters long. I've blanked out the legs, so the legs hang off down there, but they're really difficult to deal with if you leave them on. But this is the head. There's 4 eyes there, and there's 4 eyes there. Crazy. This is a bit like a giraffe with half of its eyes halfway up its neck. This is another one of the same family. It's got some horns out the front. This has eyes there, 2 there, 2 there, 2 there, and 2 there. These are just the males. The females are much more sensible. They're just normal. But the males crazy. So they're mainly in this family. There are a few that have really exaggerated heads, and these were perhaps the most exaggerated of all. Again, I've done quite a lot of processing. I've moved the scale bars up, I've taken off the legs, I've blacked out the dirt on the specimen stub, which is what we put the specimens on. So that's just an idea of the heads of spiders. Now we come to what I keep talking about, the sexual organs. So this is a very common spider. This is the female, and the female sexual organ is called the epigyne, and this is the male, and the male organ is called the pedipalp. Now on the spider, the female organ is lurking somewhere around here, so at chest level, so it's at the top of the body, below the head. And on the male, it's actually got two, and it's waving them in the air. So anyone who's looked at spiders and said, that hasn't got 8 legs, it's got 10, the top two, nearest the head, are actually the pedipalps. Now females have pedipalps as well as males, but the males develop the ends of the pedipalps into these extraordinary shapes. Like that. You can see there's an awful lot of detail there. And it was really looking at the male organ and saying, I wish I could resolve that through my light microscope. I thought, I need an electron microscope to do this. And it really brings out the amazing shape. Now, the other thing about the sexual organs is you sort of look at them in pairs, the male and the female. Because when the male inserts his pedipalp into one side of the epigyne, he's got to hold on. And he's got to inject the sperm in exactly the right place. Now, the thing about spider reproduction is that the male will inject his sperm into the female, and some days later, the female will shoot her eggs out of the oviducts, which are in there, and as they come out, they will get fertilized. So the male has to store his semen in a side canal on the female. So basically, my theory is that this here is to block off the main oviduct and direct the tube, which is probably somewhere, not sure I can see it on here, into the right place to store the sperm in the female. And a lot of people say it's like a lock and a key. So this male has got to fit onto this female so that it blocks the oviduct and injects the sperm in the right location. So with different anatomy on different spiders, you need a different lock and a different key. It is a theory that is bandied around, as we say, but it's really not a very good theory, but it gives you an idea of why there should be differences. Here's another species, just to illustrate the epigyne looks a bit different, and the pedipalp again is different. And these are just 2 more pedipalps. Now these are again 2 of my favourite pictures. It's got this enormous great hook, and it's got all sorts of things sticking out in all directions. And I think that is actually where the sperm comes from. But anyhow, this is for grabbing onto the female in exactly the right place and making the sperm goes in exactly the right place. This one is completely different. There's no grabbing on with this one. In fact, I have got a picture of the female sex organ on this, but frankly, it hasn't got one, or rather it has, but it's below the surface of the female. It isn't the case of grabbing on, it's much more a case of just injecting sperm in through here. So we call Segestria primitive spiders, and for those, the only way you can tell a female from a male, well, from being a female, is you can't see an epigyne. So it's either a female or it's immature, but you can't see any female organ. So the sexual organs were very interesting to me, but the legs are something else that they're just too small to see under a stereo microscope. And you might say, well, why not look at it under a high power microscope? But the trouble with a high power microscope is the depth of field is very small and the working distance is tiny. So if you tried to put that under a high-power microscope, you squash it with the objective lens, which is the lens nearest the subject. So you can't really do that. What you can do is you can use what's called an epi-illumination microscope, where you have the light at the same side as the lens, as opposed to looking at it from the bottom. But again, depth of field is such an issue, it's just not worth doing. So this is tarsus, which is another way of saying the feet of a spider. It's got 8 legs, so it's got 8 feet. And every spider has at least 2 of these comb-like claws. And only spiders that hunt with a web have a 3rd hooked claw at the bottom. And they also have these barbed spines. There's one there, and there's one there. And what happens is, I don't know how they do it, but they manage to grab silk, which they've spun with the hook, and lay it across these barbs, so it's like you stretch some string out and you can grab hold of it, which is really extremely interesting. And these things, as I say, are just too small to see with a light microscope. And so when I started looking at it through a scanning electron microscope, it really came to life. Just in case you hadn't worked it out, there's the hook and there's the barbed hair. Now, I talked about collaboration. Another friend of mine, who at the time was president of the British Arachnological Society, asked me if I'd like to image a spider for him because he's doing research and he thinks he'd found a new species. So this is again a foot, the tarsus, at least that's the tarsus, and this is the next bit up. And the interesting thing about this particular family is it has this strange thing here which looks like a squid, sea creature. And this is actually called the tarsal organ. Now, only this family of spiders seems to have it. And so I was able, by first finding this tarsal organ, because again, it was too small to see with a light microscope, and also looking at the male sexual organs, that he decided, yes, indeed, it was a new species that hadn't been recorded before. It's found only in the Falkland Islands, and so no, I didn't go and collect it. He collected it on a trip from about 2010, and it was only about 2020 that he got round to asking me to work on it. And it was a very scary thing to do, because he'd given me one sample because he had perhaps one other. So I had to work on this one sample and be incredibly careful. And I still managed to break one of the pedipalps, but it gave him the answer he wanted. Now, spinnerets. I showed you a picture earlier of spinnerets, and this is another species of spinneret. And under here, we've got something else. What we're looking at is this. And these are very, very fine spinnerets. That's what they look like at higher magnification. Now, just to put things in perspective, each of these spinnerets is less than a micrometer in diameter. A micrometer is 1,000th of a millimeter. A human hair is 60 micrometers approximately. So these are a lot finer than the human hair. But not only that, they are hollow and they produce silk. And the diameter of the silk is 15 nanometers, which is basically very, very fine. And what happens is the spider will eject silk from all these glands, and it will come out. It'll all be tangled up. And then it'll comb it out with this comb. There's one on each rear leg. And it will make a sort of fluffy ball of this silk. And this ball it will put round its hole in a wall where they live. And this ball of silk is sticky. And hopefully insects will climb onto this ball of silk and get stuck and the spider will come out and consume them. Moving on very quickly, there's something called a pseudoscorpion, which is a bit like a scorpion. It's an arachnid, which is the same family as spiders. They are harmless, and this particular species, which I'll show you in a minute, is found under the bark of trees. Again, they're quite small, but they're something you can just about see with the naked eye, and they make very good pictures. They have these menacing looking pulps, as they're called, but what it doesn't have is a tail that, like the real scorpion has, which in real scorpions, which are larger than these, it inject poison and so on. But these are totally harmless. The other picture I'm going to show is of a deer tick. A tick is a blood-sucking creature, and it has a mouthpiece which injects into the skin and sucks the blood from under it. And the thing about ticks is they can spread disease. And Lyme disease is a nasty thing you can catch if you're not careful. And mainly they go for hedgehogs and dogs and things, but humans can catch it too. This is what the tick looks like underneath. This is a female. It has an epigyne and an anus. And this is what's called the hypostomy, which is where it does the blood sucking. And that's what it looks like in detail. So there's a little groove here, and it sucks the blood up through the groove and takes it, I don't know, I think it probably takes it in through that hole or possibly that hole. And it extracts the interesting part from the blood, and then it regurgitates the excess plasma or fluid and puts it back into the host. And it's this regurgitation that transmits the Lyme disease bacteria from the tick to the host. When I first did one of these, a lot of these barbs were broken and the other barbs were filled with dead skin from a host. But I was lucky to find a complete one, and I managed to clean the dead skin off. So I rather like that image. It may not be to everyone's taste, of course. Right, tardigrades. Tardigrades are the species of the year. And one reason they're the species of the year is it was 250 years ago this year that someone first discovered a tardigrade. They're also called water bears or moss piglets. They're also called pudgy-wudgies, according to someone. They live in moist environments, such as moss on the lawn or on the roof. They are incredibly robust. They've been sent to space, and I don't mean inside a air-conditioned capsule. I mean just in raw space. I say they came back alive. About 60% of those that were in space came back alive on this round trip. Their normal lifespan is about 3 months, but if they find themselves in a very hostile environment, they'll sort of turn off their body clock and reduce their metabolism by a factor of 10 to the 5th. So there's 0.01% of their normal metabolism. That's 10 to minus 4, sorry. And that, in that state, they can live for literally years. Anyhow, there's lots of SEMs of tardigrades around, but I thought I'd show you a short video of a real tardigrade that happens to be alive. I just made this on Tuesday at home in my lab with a light microscope, and I'm looking up from underneath, so you can see some of the time it's got feet that it's pressing around on the bottom of the tray it's in, and you can see that it's. I'll just run it once more for fun, if I can. I showed this to someone earlier today and she was absolutely fascinated, so I thought I'd give you the benefit of an extra showing. So they're called water bears, and you can possibly see why they might be called bears. They're called tardigrade because that comes from two words, tardi being slow and grade meaning walking. And actually I think that was walking quite fast. Anyhow, that's well, it's not a bye-bye, it's a bit of interest. But I decided I wanted to look at tardigrades, and they're actually less than 0.5 millimeter long, so they're actually very difficult to prepare. In fact, they're also quite difficult to find. So I thought I'd do it the easy way. I ordered 100 tardigrades from a supplier of tardigrades, because there are such things. Well, this particular supplier sent all sorts of things to schools and labs, so it would breed fruit flies and tardigrades and all sorts of other things for use in education. So at least I had a few to play with. That's the back view of one. I found this one in my lawn, so this is one of the ones I actually prepared. I didn't buy. The head's up here. It's got 1, 2, 3, 4 pairs of legs and a number of claws, and the next picture shows the claws a bit better. This is a different species. This is one of the ones I bought, which normally live in Africa. So you can see the claws quite easily. The head is sort of face down there. I like this one. It's sort of about to pounce on you. This is actually a composite of 3 different photographs. The reason for that is, even with the enormous depth of field of an SEM, sometimes parts of the image become out of focus. So I took 3 photographs, 3 images at different focal distances, and then I combined them in software. In fact, I also did that one for that particular picture of the area where the stink bug scent evaporates, the big one which I said I liked so much. So that is tardigrade getting bigger. This shows the mouth quite well. So they have this sort of petal-like structure, and you can look inside. Now tardigrades will eat algae, so they can be vegetarian, but they can also eat things like nematode worms and things, so they do eat other animals as well. This is a close-up of a mouth. You can see it's quite menacing. There are all sorts of tooth-like appendages, and you can sort of see down the throat, and just hope you don't end up down there. And there's one at even higher magnification. For those of you who know anything about SEMs, I could say I use what's called the in-beam secondary electron detector for those last 2 pictures, which means you can put the specimen quite close to where the electron beam comes out and avoid some of the distortions from electrons getting in the way. As you can see, the scale bar, which comes in useful, is 2 micrometers, so that is actually quite high magnification. I think the magnification number I used was about 30,000. Right, well, we're just about at the end, but which is very convenient because we've had just about an hour. But the only thing I just now want to show you is I've done some work on pollen. Now I'm just going to show you 12 slides, and I've put them on a video so you have 6 seconds for each one. So you've only got 2 minutes left. On the left, I've got what's called dehydrated pollen. Dehydrated pollen is pollen as it exists on the anthers. This is the male part of the flower. So the idea is that the insect will rub up against these anthers and collect pollen, which looks like that. When the pollen is transferred to the stigma, which is the middle bit, the female bit in the middle of the flower, hopefully on a different flower, because you don't want to self-fertilize, it will swell up. Now I have taken some pollen like this and made it swell up by putting it in water. But you get the idea. So I'll set this running and I'll just let it run, but I might add a bit of commentary in between. Is it running? Yes, it is running. You just have to hope it does change. Okay, yes, it is changing as well. So this is just, as I say, this, I think it's 12 species, it might just be 6. And there's one that I will stop at because it is most interesting. So I had great fun going around the countryside collecting flower heads from these various species, and also great fun identifying them. Obviously things like holly I would know, but some of the wildflowers I didn't know before. And some of them I really can't tell apart. Right, this is one where I'll stop for a moment. Orchids pollinate in a strange way. I thought I stopped it. See if I can go back. This is really infuriating. I'll tell you what I'll do. I have a copy of that picture here. Lucky, I've actually got two pictures close to each other, which are just the ones I wanted to talk about. On an orchid, instead of having lots of these anthers, which are little male bits with pollen on, it has two things called a pollinarium. And on each pollinarium, you have a whole number of what's called a pollinium. So these are all pollinia. And when the insect lands on the orchid, it'll take, this is sticky, and it will stick to the insect. So you'll have half the pollen from the orchid will go at a single time as this complete thing. And the pollinium, even these are pollen grains, so even a pollinium, it has a whole load of pollen grains on it. So rather than individual pollen grains being taken by the insect, it takes the whole lot at once. Something I didn't know before, and I was obviously fascinated with. Now, since I'll just move on to the next one. The interesting thing about this particular species over here, well, it happens to all species, but I just happened to catch it in the act, is that here the pollen grain is passing what's called a pollen tube into the female part of the plant. And what happens is this pollen tube will go down the tube of the stigma, as it's called, into the base of the flower, and a sperm which lives inside the pollen will then migrate down into the base of the flower and fertilize the egg, and there are some nutrients as well which are passed down. So this is actually caught in the act of developing a pollen tube. Now all this might look like it's sending tubes out in all directions. It's only the area where it lands on the female plant that actually puts on down the pollen tube. So I think that is actually a very good time to come to a conclusion. Yeah, just say one thing. I'll find the right. If you want to see any more pictures of my images, that's my two websites, one for spiders and one for other SEM things, or simply just Google me, just in case I've inspired you to look further. That concludes my presentation. I'm very happy to take any questions you may have. I'll try and answer them. Thank you very much.
So thank you very much, Jeremy, for this interesting presentation. And now we have time for questions. I would like to be the first, because I noticed that you kind of like spiders a little. Do you have any of them at home? I mean, voluntarily, or do you just collect them outside?
I have about 600 spiders at home, but they are all dead, and they're all preserved in alcohol. So I obviously have a few live spiders, and I don't kill them or anything, but well, I might if I want to take pictures of their sexual organs. But I don't breed spiders. I don't mind it, but I just don't have the time or the space.
Okay, thank you. I'm glad that you live in United Kingdom. Okay, thank you.
I should say that they all live in tubes in my spare bedroom, so any visitor who comes will have dead spiders for company.
Great invitation. Okay. Okay, do we have any questions? Yes.
I just want to ask, you mentioned a few times the preparation of the specimen. Can I ask how you clean them? Because I imagine you do not have some tiny little brush to dust them off. How is this process working?
I will just repeat the question for the microphone. So the question was, how do you clean the samples? Easily.
Right,
Yeah.
that is an extremely good question, and I won't say I was anticipating it, but I have something that might illustrate the difficulty. Apart from the first slide, of course, the first picture of all showed a very dirty specimen. Right, this one. So yes, you're right. I use a little paintbrush, a tiny brush, artist's paintbrush, and I look through. I mount the specimen, then I look through my stereo light microscope and look at it and see, is there any dirt? And then I get my tiny paintbrush and I try and brush the dirt away. And that is quite hard, but I do my best. I try not to break the specimen, but that often happens. The reason I pulled up this picture is, if you look around here, it's losing its hair. It's going bald. That is exactly the problem I have. You brush away a bit of dust and you brush away some of the sensitive bits of the spider itself. You might not have noticed if I hadn't said it, but now you can see that it is definitely suffering. I still got it on the calendar, but that does illustrate the great difficulty, and preparation is a real problem. And I have to remind myself to check that they are clean, because if you put it in the SEM when it's dirty, you see it's dirty, you have to take it out, clean it later, put more gold on it, put it back in. All it takes time. It's better to get it right first time. But yes, it's difficult. By hand with a brush. Or sometimes I use a little acupuncture needle. Just if it's stuck to this, if it's stuck and won't brush away, I try and needle it away with a needle. I try not to do it afterwards in Photoshop. Yeah, so it is, and sometimes you ruin the specimen while you do it. It can't be helped.
Okay, I have a related question. So do you gold coat all your specimens, or do you also use low vacuum molds?
Sorry, can you speak a bit louder?
Do you gold coat all of your specimens, or do you use the low vacuum molds too?
Just if you can repeat the question to the microphone. Okay, it would be me. If you put gold on all of the samples.
Right.
Yeah? Okay.
Short answer is almost all my specimens I coat with gold. And for people who wonder why I coat with gold, it is to avoid an electric charge building up on the specimen and interfering with the electrons that are emitted from the specimen. So it's to make sure you have a good picture. And the question was, do I use something called low vacuum mode instead? Low vacuum mode is a way where you actually have a little gas in the chamber, and that will counteract any electric charge. The gas gets positively charged, and the positive charge goes to the negative charge on the specimen and cancels it out, so you don't get the trouble with excess charge. I almost always use gold. I do have low vacuum mode, and I sometimes use it. Actually, I use it more with mineralogy than I do with insects, because minerals create all sorts of problems. I'm just starting to look at minerals, and I'm learning all the difficulties. So low vacuum mode is that. I have used it. One thing, I haven't got a picture here, but if you have an insect and it's got antennae that are crossed, the gold won't necessarily get to the antenna that's underneath. So there's a small part of the insect that isn't coated with gold, and that will charge up, or the bit, it'll stop the charge escaping, and then you can get artifacts on the picture. So you reminded me, I have one very picture that I like very much, but I had to use low vacuum mode to avoid that effect. But normally, just gold coating.
So you mentioned it's quite an expensive thing to... The microscope is quite an expensive thing, you mentioned. But what about the maintenance? Can you have some is it expensive to run it? And also, do you need some help from the manufacturer to run it sometimes to fix something replaced?
Right.
This is actually a good question because we have Teskan here. Okay, the question is about the maintenance of the electron microscope, if it's expensive and if you need any help from the manufacturer.
Right, yes. This is not a cheap hobby. When I bought my first one, my second-hand electron microscope, I bought it from a man who runs a company who sells second-hand equipment, and he maintains equipment from any manufacturer. He said, "Do you want a maintenance contract? Do you want to pay me on a visit-by-visit basis?" And I asked how much was a maintenance contract, and I decided I'd do it on a visit-by-visit basis, because each visit would cost me £1,200, something like that. Quite a lot of money. And it didn't break down very often. But there was one time when it did break down. And to cut a long story short, it required a new or reconditioned high voltage power supply. That cost me as much as a small car to replace, because I actually then had to get the manufacturers of the microscope in to put an exchange unit in, because my engineer could not get hold of one, and anyhow he didn't want to get one and then find it wasn't right and have it sent back. So my engineer was very glad that I did not take a service contract out. Coming back to the present, the MARA, yes, I shall have a self-service contract. Among other things, it means that they can update the software every time there's a new software release. Yes, it's expensive. I won't say how much. But it is, while I like collaboration with people who like insects or spiders, I also do love the SEM as an instrument. So I do love the collaboration with Teskan as well. In fact, it's one reason I'm here, that we get on well, we enjoy what we do, and we feel like a team. So yes, it is expensive. It's not a cheap hobby. You can play a little dangerous and not take a service contract out. You can buy second-hand equipment and hope, but you've got to be prepared for a big expense. It's not something you can mend yourself, really. Sorry.
Thank you for the amazing lecture. I would like to ask, does it happen to you sometimes you feel the urge you need to prepare a focused ion beam like lamella to examine the cross-section?
Do you want to repeat the question?
I can't repeat it, but okay.
The question relates to focused ion beam applications, whether you have a temptation to cut with FIB and to see what is underneath.
Yes. I mean, no. I understand the question. Focused ion beam is where you use a separate electron, well, it's not an electron beam, it's an ion beam, which is more power, to eat away at the specimen, so you can effectively remove some of the specimen, take an image, and remove a bit more of the specimen and take another image. I don't think I'd use one an awful lot. If I had one, I would use it and I would make images, but we are talking hundreds of thousands of pounds, and I live and work on my own. I'm a retired engineer. Okay, I have some money in the bank, but actually they take up more space as well, and my lab's a bit small. So yeah, it's a nice idea. What I will say is I recently started getting interested in EDS, electron dispersive spectroscopy, which is basically a way of working out what something is made of. Now with spiders and things, we know it's carbon and oxygen and hydrogen and things like that. But when you're looking at minerals, it's a very good way of determining what the mineral type is. And I'm just getting interested in minerals with a couple of professors I'm working with. And so I have an EDS system on loan at the moment, and I'm talking to Teskan about which is the best system to buy. So I am spending money on EDS, but that is somewhat less than FIB. Any
other questions? Well, If not, thank you very much for your questions, and thank you, Jeremy, for the answers and for the whole lecture. Ladies and gentlemen, this is the very end of today's evening. Thank you very much for coming. If you're interested about some other events during the Electron Microscopy Days, you can check our website, which is dem.brno.cz, and you can find also very interesting events. There is a lecture tomorrow and a program for families on Saturday, and so on, so on. We've prepared really great program. Yeah, I have to say it. So thank you very much for coming, and hopefully see you again, and have a nice rest of evening. Goodbye.